So good afternoon, everyone. Today I will share a little bit of our work, part of my PhD, insight into SHP2 Latch allosteric site, advancing covalent allosteric inhibitors. So as you heard from Nina, essentially SHP2 is a protein tyrosine phosphatase, BUC3 expressed everywhere in our body, and it's a cytosolic protein. Particularly, it's particularly, it's a downstream of tyrosine kinase receptors, regulates cell adhesion and migration, and that's why it's also important for macrophages and also T cell activation. In addition to this, mediates several responses down in the cell line. So it exists in a closed basal autoinhibited state where the NSH2 domain, so to answer Patrick, engages the PTP domain, where there is the catalytic activity, and mutation generally leads to activation, inhibition of the autoinhibited closed state, and activation of SHP2, leading to enhanced basal activity. So there are three allosteric sites, and one of that is actually, I didn't face where Patrick was looking for. One is the tunnel site, the latch site, and the groove site. On the groove side, there are not so many, no structure available at the moment. Actually, there is even a cryptic pocket on the SHP2, which has been identified. On the tunnel side, there are a plethora of compounds, and it's been at the focus of research efforts in the last decades. Why? Because one of the first compounds synthesized was showing the ability to be able to active and omit mutants of SHP2 compared to the orthosteric site, orthosteric ligand, which is actually not, it loses activity. So we decided to focus on the less explored latch site, for which just few papers are available. And essentially, we started our campaign considering these Novartis inhibitor, non-covalent inhibitors. They were published in a paper where they also demonstrated dual allosteric inhibition when tackling both the tunnel and the latch site at the same time. So how to start a covalent campaign? Of course, we have to consider the binding site and the residues available in this binding site in order to decide which warhead to use for targeting. In this case, the choice was quite easy because these non-covalent inhibitors are already able to engage the lysines present in this site. So we just decided to focus on improving this inhibitor even more through the covalent strategy tackling these lysines. How? Introducing warheads, equipping warheads, installing them on either the proline moiety or the aromatic moiety here bonded to the triazole. So these are the first results. These first molecules were supported partially by computational docking. And essentially, you can see on the top the modification of the proline part with the covalent warheads, and on the bottom with the modification on the aromatic part. However, these initial results were not that promising because, as you can see, we couldn't actually beat the activity of the inhibitors, non-covalent inhibitors, maybe except in the case of the 4-hydroxyproline and in the case of these dehalogenated methyl pyridines. However, we were parallelly doing covalent computational investigation to identify which key interaction may be playing a role in even the not engaging lysine compound of Novartis. And through dynamic undocking, some interesting interaction that may be otherwise underestimated were identified. And together with this, we also conducted a screening, experimental screening of an in-house lysine library. 39 compounds were tested, of which 28 showed an inhibition above 50% in a 10 micromolar concentration, Then of these, we conducted IC50 measurements. 17 were selected for their promising IC50 and thus tested for intact mass. From these 17, 7 that you see here showed to be able to tackle the lysine in the latch and also in the tunnel side. And that's what we were looking for. Particularly a deeper analysis, a closer analysis, I think you can all see that essentially 3 out of 7 present an NHS ester as active warhead, essentially. So from this, the idea to install, even on the heat compounds in the Novartis compound, this moiety for reaching a labeling of this lysine. The first compound we modified was the SHP2 scaffold, SHP244 scaffold. Essentially, we switched the orthovanillin moiety with a benzoic moiety and installed the NHS moiety. As you can see, we got already some promising results, still in the low micromolar range. But if we compare it to the non-covalent analogs, these are quite promising. And especially if we compare to the other non-covalent inhibitor of Novartis, we can see that the loss given from not installing a phenol, an OH on the aromatic ring, can be rescued and even overcome by the covalent strategy. We suppose that probably at the base of the efficacy of these compounds, there is actually an interaction, an H bond with the lysine 2 and 66 from this side of the molecule. Following these promising results, we also continued our investigation. I'm sorry that you see the black square there. With SHP844, we modified this molecule, and luckily we could reach much promising compound. We reached nanomolar activity, and you can see there is a difference in the stereoisomers, particularly the S isomer, which is the same as SHP844, is actually very strong inhibitor from this point of view. Why? We think in this case, it's the proline is allowing a closer proximity to the lysine. What? Sorry, I'm sorry. I clicked. I'm sorry. Yeah, a closer proximity to the lysine 274, and again a nitrogen bond. It's allowing the interaction and the covalent mechanism to take place easily. Interestingly, the non-covalent analog, the R stereoisomer of this SHP844 at 10 micromolar, so we don't know for higher concentration how it is, but at 10 micromolar didn't show any activity. This led us to the idea that maybe the proline is actually just disposing closer the carbonyl in the case of non-covalent inhibitors and the NHS ester in the case of the covalent inhibitors to the lysine. So it actually is not probably that much essential for the activity. To test this hypothesis, we decided to test one of the actual intermediate in the synthesis for SHP844 compound, this carboxylic acid, which has also the requirements seen before in the dynamic undocking, the key interaction. And luckily, we could see a very promising activity, actually even overcoming the non-covalent inhibitors of Novartis, even though this is still a non-covalent inhibitor. Thus, we proceeded with the installation of an NHS ester to verify if we could achieve a stronger potency. However, in this case, the activity was not increased. We suppose it's because there is a bigger distance from this side from the lysine. So even the NHS ester is not able to engage them, and thus the covalent mechanism does not take place. Thus, the idea to use one of the other 7 compounds that showed to label the lysine and to use some small linker to favor the elongation and the reaching of the lysine, which of this was supported by computational investigation docking especially. And we reached another much promising compound, this dichlorotriazine derivative with an hydrazine linker, which could actually engage. These are still docking structure, but we are proceeding with X-ray structure determination, which could actually achieve a closer proximity to the lysine. And it's more interesting also from medicinal chemistry point of view because of the mechanism of action, which is different, and the stability too of this moiety compared to NHS esters. Interestingly, also in this case, the non-covalent analog, the hydrazine analog, is much less active. So I hope that with this presentation, I could show you that fragment screening is an important experimental approach that it can actually help in the tuning of covalent inhibitors, which is not a straightforward process, but it requires carefully optimization and computational and experimental investigation in the process. NHS ester, however, are promising chemical biology tools, and they can use to identify lysine, which may be tackled by covalent inhibitors. However, there are still limitations in this whole process, and essentially the synthesis, it's a very long, complex, and it's actually limiting the research on this side. So synthetic efforts are required, but the good news is that I hope you could see that it's possible to reach a good activity even on this side that has been a little bit underestimated from the research community so far. And we are proceeding with further modification, maybe taking into account other compounds. Thank you for your attention, and thank you all for the ALLODD consortium, my group in Budapest, and thank you for your attention again.
Thank you very much. Hi, thank you very much for your presentation. Congratulations for the result of your team. It is more curiosity than a real simple question, I think, but I've seen the design of many covalent inhibitors, and most of them are micromolar, and a couple of them are nanomolar.
Yes.
So, what is your explanation for this? Is it non-covalent, essentially, or and how do we make sure whether a covalent inhibitor is?
Yes, let's say that when you install a moiety warhead, the covalent mechanism is not as straightforward process as said, so it is not always sure that you get a covalent compound after installing a warhead. Depends on the reactivity of the warhead, depends also on the interaction that the warhead is actually establishing. In that case, we think that probably they are not covalent compounds because you see an increase in activity when, of course, a bigger increase in activity when they actually are covalent. But yeah, there is this little bit idea that then you install and you get a covalent, but unfortunately, it's much complex than that. Apart from synthetic point of view, that you can imagine the challenges sometimes to not using any nucleophilic solvents or even water sometimes. Depends, of course. There are more stable warheads, there are less stable ones. Depend on that. Please.
Thank you, Vincenzo. It's always nice to hear the progress on your perspective on this chart curtain. I was wondering, for the lysine residues in the Latch side, do you have any pKa calculations or solvent accessibility that would, or that maybe allowed you to prospectively estimate their reactivity towards your warheads?
So we did some calculation and the simulation in Barcelona in the secondment. Actually, we haven't finalized that because we were already getting some promising results. These are solvent-exposed lysine, so the pKa is generally estimated to be higher. However, even this lysine, even when they are solvent-exposed, doesn't mean that they are not, they cannot be tackled by covalent mechanism. And this is one proof, but there are other literature references. Of course, they are much less nucleophilic. So we suppose that at the base of the interaction, there might be a need for interacting with the lysine, maybe through the hydrogen bond, as in the case of NHS esters. But still lysines targeting let's say it's more complex than a cysteine targeting, but it's the progress are being made, and hopefully more secure, stable, and effective compounds and strategies can be done in this way, and maybe our research can also contribute in that. So yeah, I hope I answered. Thank you.
My question is the following. Have you measured the lifetime of the metabolic lifetime of the protein in vivo? And is there how many hours or days the protein is stable? And does the covalent change eventually the lifetime of the protein?
No physiological studies have been made, just in vitro study, biochemical studies. We were thinking to proceed with cell studies to verify, but no physiological data were conducted from our side. Probably in the literature there is some reference. I'm not sure of that, to be honest, at the moment. But yeah, maybe it's something to look into. The covalent compounds and moiety, depending on the moiety, they can be more or less stable in a certain buffer, and thus also you can imagine cells in different situation. They can hydrolyze or not, or actually be stable enough to engage these compounds. Depends. It's very variable. It's very one cannot say in advance. Sometimes it's also a matter of interaction sometimes. So it's a very interesting point. This is just a start from like trying to pave the way for interesting activity also on this side, shine a little bit some light on this side that has been a little bit underestimated. Thank you.
For me, a question to you also, because you have the Latch side and the Athens side.
And the group too.
Why Allosteric inhibitor, which tackles both sides? This is possible, in my opinion, and maybe a good strategy.
You mean one compound that could tackle both? Well, I think there are some differences in the composition. That's maybe also the inhibitors have a difference in structure. So it might be possible because the Latch side, it's probably possible because the Latch side is solvent exposed, so maybe it's easier to reach. The tunnel, maybe too. There has been no effort in this sense for now, but yeah, we were focusing on trying to optimize this as starting point for sure. How far are they? That's a good question. I think they are quite in different sites, right, Nina? Yeah, they are quite far. Yeah, they don't share any residues in common, let's say. Maybe this one.
Compounds combine simultaneously on the tunnel side. It's just what Vincenzo said, that the properties on the are quite different. So if one compound that recognizes both can be developed mature, but they can be targeted at the same time.
Okay.
Yeah, that's why they developed this in this paper, actually dual allosteric inhibition. Like they use one Latch side and one tunnel side compound, and they test it together. And I think it's more efficacious this way, maybe for them.
Okay, thanks a lot.
Thank you.